Assalamu alaikum. Today we will discuss what is molecular docking studies. Molecular docking studies is actually in silico ligand target interaction. In silico means computer based ligand target interaction. So first of all we should know what is ligand. Ligands are small molecules. They are also called as signaling molecules, may be organic or inorganic in nature. Today we are going to use nanoparticles also as a ligand. Small peptides or cyclopeptides can also be used as a ligand or the segment of DNA or RNA, and sometimes metal or metal complexes may also be used as ligand. How we can design our ligand? We can obtain ligand from different sources. First of all, we should know what is co-crystallized ligand. Co-crystallized ligand is actually the crystal structure of the ligand which is complexed with the target, that is ligand bound with the target, and we can obtain its crystal structure from X-ray crystallography. Ligand obtained from library. We can obtain different ligands from different libraries like zinc database. We can use PubChem, Drug Bank, ChemSpider, and we can also draw the structure of our ligand through ChemDraw, or some other tools can also be used. There are some online tools available which can be used. And energy minimization is also important. So before our molecular operating environment, these two softwares can be used to energy minimize our ligand. The second thing is the target. Target is actually the macromolecules or large molecules, which are also called as receptors. They are proteins, maybe sometime enzymes, DNA, RNA, or polymers can also be used as a target for ligand interaction. To obtain target structure and processing, we should keep in mind that three-dimensional crystal structure and proper folding of our receptor is important, and we can obtain this structure from protein data bank. I have made this video that how we can obtain 3D structure of our target from protein data bank. So you can visit my YouTube channel for that video. Uniprot or Swissprot can also be used. And for processing of the target or receptor, we should have to do some modifications like protonation, removal of water, and energy minimization to finalize our structure for molecular docking studies. Our different softwares use different interaction models, but most of the softwares are based on log key model. So on the basis of log key model, most of the softwares work. Now there are some other and recent softwares available which have an option of induced fit model, and we can use induced fit model during docking studies. How we can analyze our sample, or after docking, how we can predict that what is the our results, what are our results. So there are three important factors which are important, like type of interactions. We should know what are the different kind of interactions during ligand target interaction, binding energies, and RMSD value. So these three are the important factors during analysis. The first thing is the type of interaction. The detailed video of type of interaction is also available on my YouTube channel. So there are different type of interactions like metal-ion interaction, conventional hydrogen bond, halogen bond, pi-pi interactions, pi-alkyl, pi-cation or anion interactions, and there are some other hydrophobic interactions. The second thing is binding energies. Binding energies are important. These are the minimum amount of energy to stabilize ligand target complex. So these binding energies are also called as Gibbs free energy, and these are the sum of all the interaction energies during ligand target interaction. RMSD value is the root mean squared deviation. The best fit value for RMSD is 0.1 to 2 angstrom, And RMSD can be obtained by overlapping atoms or heavy atoms of our ligand with as compared to reference ligand. And the reference ligand can be obtained by we can by using co-crystallized ligand. So co-crystallized ligand can be used as a reference ligand. There are some softwares which automatically use or calculate RMSD values during docking studies. Virtual screening is a type of molecular docking study which is based on uh, two different types: ligand based virtual screening and structure based virtual screening. So in ligand based virtual screening, ligand structure is important. So we have an active compound. On the basis of that active compound, we obtain multiple ligands or library of compounds, and we are going to dock in the enzyme or the target. While during structure based virtual screening, the structure of the receptor or the enzyme is important. That is active site, what kind of residues present inside the active site, and what kind of catalytic mechanism it is going to use. And on the basis of that, we are going to design our different ligands, and then we dock our ligands inside the receptor. Different softwares can be used for molecular docking. Lot of softwares available. I'm going to examples. Some examples are given here like Autodoc, Pyrex, Gold, MOE. MOE is Molecular Operating Environment, Discovery Studio Visualizer, DSV, or Glide. And there are a lot of other softwares we can be used. It depends on availability of the software, and also online softwares can be used, like SwissDoc is an important online database or software which is available and can be used for molecular docking. Applications of molecular docking. Molecular docking can be used to predict structure activity relationship, ligand receptor interaction, and more specifically, we can say that enzyme inhibitor interaction. So, inhibition of the enzyme can be studied. We can calculate the energy values during complex formation, and we can also screen library of compounds during molecular docking studies, and we can obtain hit compounds. And these interactions or molecular docking studies are also important during drug discovery process. Thanks. If you like my video, please subscribe my channel and share this video.